A very good morning. I am Lakshminarayana Gunta, PGT in Zoology at APMS and Junior College Garbanja, and also working as NEET Super 60 Zoology faculty at Dopulvalsa, Srikakulam. Today we are going to discuss the physiology of excretion. In our previous segment we have completed the first step, that is the glomerular filtration. Today we are going to discuss the other two steps, that is the tubular reabsorption and the tubular secretion. We have seen that the glomerular filtrate is equal to plasma minus proteins and fats. Means the glomerular filtrate is plasma without proteins and fats. That's why 99% of the material that is filtered is reabsorbed by the renal tubule. As a result of this reabsorption, the urine contains mostly the waste materials and excess of water. Okay, this tubular reabsorption, it occurs in all the regions of the renal tubule. Most of the reabsorption, it takes place in the proximal convoluted tubule. And coming to the tubular secretion, this tubular secretion and tubular reabsorption, they differ in the flow, in the direction of transportation. Here in this diagram you can observe, this one is the glomerular filtration, the first process in urine formation. Coming to the second one, that is tubular reabsorption. In tubular reabsorption, what is happening? The fluid, which we call it as glomerular filtrate or tubular fluid, it moves. Most of the contents of the tubular fluid, that is water, glucose, amino acids, they move from the renal tubule to the peritubular capillary. This is the flow of direction of tubular reabsorption. Whereas coming to the tubular secretion, here you can observe the excess substances, they are sent into the renal tubule. Means tubular reabsorption, it takes place from the renal tubule to the peritubular capillary, whereas tubular secretion, it takes place between the peritubular capillary to the renal tubule. Okay, both these processes, they occur simultaneously. Okay, that's why we also study these two processes at a time. Okay na? In tubular reabsorption, the substances, they are transported from the lumen of the renal tubule, which we call it as deletion. Okay, they are deleted and they are sent into the interstitial fluid, and from there they are sent into the peritubular capillary. And whereas in tubular secretion, the substances, they are transported from the interstitial fluid into the tubule, into the lumen of the renal tubule, which we call it as addition. We are adding substances to the tubular fluid. That's why it is called as addition. Okay. Most of the filtrate collected in the tubule, it is reabsorbed into the blood through these peritubular capillaries. How it happens? During this reabsorption, actually the fluid, the tubular fluid, move out of this lumen and reaches the epithelial cells. What are the type of cells present in the proximal convoluted tubule? Yes, they are cuboidal epithelial cells with microvilli, which increases the surface area. Okay, and from there the fluid, it joins the extracellular fluid. Okay, and from there finally it crosses the peritubular capillary and enters into the lumen of the peritubular capillary and joins the blood. Okay, this is the passageway for reabsorption. Okay, this reabsorption, it may be because of active transport or maybe because of passive transport. The passive transport, it doesn't involve any expenditure of energy. The examples for this passive transport are water and urea, they are transported passively, whereas glucose and amino acids, they are transported actively. Means for the transportation of this glucose and amino acids, some amount of energy is required in the form of ATP. Okay, that's why it is called as active transport. Whereas sodium ions, they are transported both actively and passively. The sodium ions present in the tubular fluid, they are transported to the epithelium of the renal tubule passively, but from there they have to move into the interstitial fluid. This happens actively, means the movement of sodium ions from the epithelium to interstitial fluid, it consumes energy. That's why it is called as an active transport. Whereas this water, it is reabsorbed in almost all parts of the renal tubule except the ascending limb of Henle's loop. Loop of Henle. This is the basic structure of an nephron, and this one is the Bowman's capsule, and this one is the proximal convoluted tubule, and this one is the distal convoluted tubule, and connecting this proximal and distal convoluted tubules, a hairpin shaped structure called as the loop of Henle, and this loop of Henle is again having two regions, the descending limb of loop of Henle and the ascending limb of loop of Henle. And this ascending limb of Henle's loop, the water reabsorption takes place. Water is reabsorbed in all the parts of the tubule except ascending limb of Henle's loop. Almost all, most of the water, that is 80% of water is reabsorbed in the proximal convoluted tubule and the descending limb of Henle's loop. Okay, this is the water here in the proximal convoluted tubule and the descending limb of Henle's loop is reabsorbed, and this method of reabsorption is called as obligatory reabsorption. Whereas coming to other substances, most of the reabsorption, it takes place within the proximal convoluted tubule. 65% of glomerular filtrate is reabsorbed at the proximal convoluted tubule, and the cells of the proximal convoluted tubule, they are epithelial cells with a number of microvilli. I have already told you these microvilli, they are essential for increasing the surface area. These microvilli, they increase the surface area and they have a brush border. This type of arrangement is responsible for the increasing of surface area, which in turn provides the place for numerous mitochondria and glycogen, which are very much essential in active transportation from the lumen of the tubule, renal tubule, to the interstitial fluid and further into the peritubular capillary. Okay, na? Let us know what are the substances they are reabsorbed. The substances reabsorbed are glucose, all amino acids, water. 80% of water is reabsorbed in the proximal convoluted tubule, and 70% of sodium ions are reabsorbed in the proximal convoluted tubule, and also 75% of potassium ions, urea and vitamins, particularly vitamin C, they are reabsorbed in the proximal convoluted tubule. Entire glucose and amino acids, they are reabsorbed at the proximal convoluted tubule. Normally, our blood glucose is 100 milligrams per 100 ml. If it is excess, then what happens? Automatically, the blood capillaries, the peritubular blood capillaries, they reabsorb the glucose from the tubular fluid. Okay, but they can reabsorb this tubular fluid only up to a threshold value, that is 180 milligrams per 100 ml. This 180 milligrams is called as the threshold value of glucose. If it is exceeded, if the blood contains more than 180 milligrams of glucose per 100 milliliter of blood, then automatically the kidneys, they excrete this urea. Our kidneys, they excrete this glucose, which is called as glucosuria or glycosuria. Elimination of glucose or excretion of glucose through our urine is called as glucosuria or glycosuria. And this glucose, it is a high threshold substance. Based on their nature of reabsorption, the substances are divided into three types: high threshold substances, low threshold substances, and a threshold substances. High threshold substances, they are almost reabsorbed into the peritubular capillary. The examples are glucose, water, sodium, potassium, etc. Okay, glucose, it is 100% reabsorbed. 99% of water is reabsorbed. 98 to 99% of sodium is reabsorbed. 100% of potassium is reabsorbed. These are called as high threshold substances. Whereas low threshold substances, urea and uric acid, they are called as low threshold substances. These substances, they are reabsorbed up to a certain extent. That's why this urea and uric acid, they are also called as primary threshold substances. And A threshold substances, A means no, completely they are not at all reabsorbed by the peritubular capillaries. The examples include creatinine phosphate, hippuric acid. These are the examples for A threshold substances or no threshold substances. Okay. Creatine and sulfates, they are not at all reabsorbed by the peritubular capillaries. And we already have discussed that this nephric filtrate or glomerular filtrate, even though it doesn't contain any proteins and fats, but it is isotonic with the plasma of the blood. Can you tell me the osmolarity? What is osmolarity? Concentration of dissolved particles of chemicals and minerals like sodium and other electrolytes. Generally, in a healthy person, the osmolarity of blood plasma is 275 to 295 milliosmoles per kg. The osmolarity of the nephric filtrate is also 300 milliosmoles. That's why this nephric filtrate, it is isotonic with the blood plasma. Okay. Coming to the tubular reabsorption and tubular secretion in loop of Henle. In the loop of Henle or Henle's loop, the reabsorption is minimum. This descending limb of Henle's loop is freely permeable to water but almost impermeable to electrolytes. That's why in the descending limb of Henle, the tubular fluid, it becomes hypertonic. Why it is hypertonic? What happened to the fluid? Descending limb of Henle, freely permeable to water but almost impermeable to electrolytes. As the water always repeatedly moves from the lumen of the tubule into the interstitial fluid and finally into the peritubular capillaries. As the water is decreased in the tubular fluid, what happens? Automatically the concentration is increased. When the concentration is increased, then it becomes hypertonic. Then the osmolarity also increases. The osmolarity of the blood plasma is 300 and the osmolarity of the nephric filtrate is also 300 nearly. Okay. And as the water is reabsorbed repeatedly from the loop of Henle, what happened? The concentration of electrolytes is increased. Thereby, you can observe here clearly what happens. The osmolarity is continuously increasing. Here it is 300 milliosmoles per kg, whereas it is gradually increasing to 500, 600. And when the tubular fluid reaches the U-shaped bend, it reaches to 1200 milliosmoles. The osmolarity is increased almost 4 times of the osmolarity of the blood plasma. Okay, but when it starts reaching the ascending limb of Henle, what happens? The ascending limb of Henle is impermeable to water, and it throws out salts like sodium ions, potassium ions, magnesium chloride ions, etc. As the tubular fluid it ascends up, almost the electrolytes they are reabsorbed. And the water it will not be reabsorbed. That's why automatically what happens? As the electrolytes are removed from the tubular fluid, then the concentration automatically decreases. Then you can clearly observe here, the osmolarity is gradually decreasing. By the time it reaches the distal convoluted tubule, the osmolarity almost it becomes 100 milliosmoles. Means what happened? Is it hypertonic or hypotonic? Yes. It is hypotonic with the blood plasma. Initially, at the Bowman's capsule, the nephric filtrate is isotonic with the blood plasma. In the descending loop of inlay, it is hypertonic, and in the ascending loop of inlay, it is hypotonic, and finally it becomes hypertonic by the time it reaches the collecting duct. Okay, the amount of fluid that enters the distal convoluted tubule per minute is 25 ml only. I have already told you that the GFR, glomerular filtration rate, is 125 ml per minute. Here, from the thick ascending limb of inlay to distal convoluted tubule, only 25 ml of tubular fluid is sent per minute. Why? What happened to the remaining 100 ml? It automatically indicates that is reabsorbed into the peritubular capillary. Means tubular fluid, it is getting concentrated. It is getting concentrated. That's why more than 80% of filtered water has now been reabsorbed. And the early part of distal convoluted tubule, here you can observe, it reabsorbs small amounts of sodium ions by active transport, by the influence of aldosterone, a hormone called as aldosterone, which is produced by the adrenal cortex. Okay, and also some amount of small amounts of chloride ions also reabsorbed, and here the potassium and hydrogen ions, they are secreted into the distal convoluted tubule in order to maintain the pH of the blood. Okay, the pH of our blood is generally ranges between 7.35 to 7.45. Okay, that is our blood is slightly alkaline. Suppose for a pH of 7.4, the number of hydrogen ion concentration may be, suppose, 40 n moles per liter. If the hydrogen ions, these H plus ions, they are 100 per liter, then the pH it is decreased. Suppose it may be decreased to 7 pH, neutral pH. Then automatically what happens? The increased hydrogen ions, they are secreted into the distal convoluted tubule. Thereby the hydrogen ion concentration is decreased inside the blood. This way, in order to maintain the pH of the blood, the hydrogen ion, they may be secreted into the distal convoluted tubule. Okay, this is all about the tubular reabsorption and tubular secretion. And finally, coming to the collecting duct. This one is the collecting duct. This one is the renal pyramid, and this portion, it is enlarged here. It is enlarged here. You can clearly observe. This one is the collecting duct, and large amount of water, it is reabsorbed in the collecting duct. Thereby the urine, it becomes further concentrated, more concentrated, and this collecting duct, it also sends some amount of urea into the medullary interstitium in order to maintain the osmolarity, and also it plays an important role in maintaining the pH and also the ionic balance by selective secretion of the hydrogen ions and potassium ions, as like we have discussed in the distal convoluted tubule. Finally, concentrated urine is sent out. That is why the urine we pass out is hypertonic urine, and the ADH or vasopressin, it plays an important role in making the urine a concentrated one. How it makes the urine a concentrated one? It acts on the distal convoluted tubule and the collecting duct for the reabsorption of water. This antidiuretic hormone ADH or vasopressin, which is secreted by the cells of the hypothalamus and stored in the posterior pituitary. Vasopressin and oxytocin, they are produced by the hypothalamus and stored in the posterior pituitary. Vasopressin or antidiuretic hormone, it is stored in the posterior pituitary in the form of herring bodies, and they are released into the blood. They reach the kidneys and they act on the collecting duct and distal convoluted tubule, thereby they promote the reabsorption of water. That's why they prevent the diuresis. Diuresis means excretion of large volume of dilute urine is called as diuresis. Tea, coffee, they are called as diuretics. Whenever we drink tea or coffee, within a span of time, we feel the urge to pass out urine because they act as diuretics. This anti-diuretic hormone, it prevents diuresis, means it promotes the excretion of concentrated urine. Due to the hyposecretion or deficiency of anti-diuretic hormone, what happens? Automatically the reabsorption of water at distal collecting tubule and the collecting duct is not proper, and that's why more amount of water is released. That's why our urine becomes dilute urine. We feel more thirsty and we may take up to 30 liters of water per day. This frequent urination is called as diabetes insipidus. We have already studied diabetes mellitus and diabetes insipidus. Mellitus means sweet. We excrete sweet urine, means sugar is eliminated along with urine. Insipidus means tasteless. Here the urine is tasteless because more amount of water is added. That's why the urine becomes tasteless. That's why this condition of frequent urination because of dilute urine is called as diabetes insipidus. Finally, the urine is formed. Let us observe the flow of urine. First of all, the glomerulus, it filters the blood and the filtrate is called as glomerular filtrate or nephric filtrate. Okay. This glomerular filtrate, it reaches the Bowman's capsule. From there it reaches the proximal convoluted tubule, and from there it reaches the descending limb of Henle and ascending limb of Henle. Distal convoluted tubule, this one is the distal convoluted tubule. Collecting tubule, collecting tubules, they unite to form a collecting duct. These collecting ducts, they open into a duct of Bellini, and these ducts of Bellini which open into the renal pyramids. These renal pyramids, they end with renal papillae. These renal papillae, they open into cup-shaped renal calculi. These renal calculi finally they open into a funnel-shaped renal pelvis. This renal pelvis opens into the ureter, and these ureters, they finally collect the urine, both the ureters from both the kidneys. They collect the urine into the urinary bladder. This urinary bladder, it opens into a urethra. From this urethra, the urine is sent out of the body. This is the flow of urine. This finishes our today's session. Thank you for watching, and follow me on YouTube simply by typing Lakshminarayana Gunta. One more thing, don't forget to take the online exam which I have provided below the video in the description box. There is a link. Simply touch that link, and for each and every topic I have provided an online exam for 60 marks each. Okay, thank you.